Hello everyone, welcome back to another video series of the sexual reproduction in flowering plants. So in this video we are going to talk about the basic changes which occur to the different parts of the flower once the fertilization event has done. So those changes are called as post-fertilization changes. So here you can see that structure which I have drawn. Maybe this is familiar to you. This is the funicle, this is the part hilum, this is the micropylar end, chalazal end. This is the structure of the embryo sac. These are the outer and inner integuments of the ovary. Rest of the part of the flowers, what will happen to them? Like calyx, corolla, androecium, rest of the part of the gynoecium, receptacle, will they fall off? Yes. After the fertilization event is happened, what will happen to the calyx, corolla, androecium and gynoecium? All of them are going to fall off. Another part, the part of androecium and except this portion, the another part of the gynoecium, all of them will wither or all of them will fall off. Now exception is there. Exception is everywhere, right? Exception is there in case of family Solanaceae. In case of family Solanaceae, we can find the persistent calyx in the fruits. Now, what is the meaning of persistent calyx? In case of brinjal, can you find that this kind of structure is present? Like in case of, if this is the structure of the brinjal, then we can find this type of structure, right? This is called as persistent calyx. But in most of the cases, the calyx, corolla, androecium, gynoecium, all of the parts can fall off, okay, or wither. So this is first change which can be seen in most of the flowers. This is the first change where we can find that calyx, corolla, androecium, gynoecium can fall off. Exception is the Solanaceae family. In Solanaceae family, in most of the fruits we can find the persistent calyx, like in case of tomato, capsicum, chili, brinjal, we can find this type of structure which is considered as persistent calyx. Next conversion, that is the ovary will get converted into ovule. So we can say that ovary will be converted into fruit, not ovule, sorry. Ovary will be converted into fruit. Alright. Ovule will be converted. The ovule will be converted into seed. Ovule will be converted into seed. Sorry, this is not the ovary. I have also drawn the wrong picture. The structure which is containing this type of structure, that is the ovule, that structure is considered as ovary. So the ovary will be converted into fruit and the ovule will be converted into seed. Now what will happen to the rest of the portion of this part? Like this is the outer integument. This is the outer integument. So the outer integument, and this is the inner integument. This is the inner integument. So outer integument will become testa, that is the outer seed coat. Okay, testa is the outer seed coat. And the inner integument will become tegmen, that is the inner seed coat. Okay, outer integument will convert into the testa, which is outer seed coat, and inner integument will be converted into tegmen, which is the inner seed coat. Now, what will happen to this? That is, this is the zygote, this is the 2N, and inside which there is the pen, or we can say that this is the endosperm. Okay, this is the endosperm. After doing the double fertilization event that will lead to the formation of the zygote and the endosperm or pre-endospermic nucleus. So from the pen, the endosperm can be developed. So what will happen to that endosperm? What will happen to that endosperm? In 90% of the plants, we cannot find any kind of endosperm. The endosperms are actually very rich in nutrients, and the function of endosperm is to provide nourishment to the growing embryo. The embryo is present, embryo is forming from the zygote. Once the zygote will show repeated division, they will form the embryo. I will talk about the embryo and how the genesis of the embryo is happening from the zygote, that in my next videos. But now, for now, just see that the zygote is converted into the embryo and the primary endospermic nucleus is forming the endosperm, which is absolutely rich in nutrient. Now, in most of the cases, while growing up of the seeds, the embryo can use all the endosperm. That is why those seeds are considered as non-albuminous seeds. But in few cases, we can find the endosperm. If this is present, if the endosperm is present, then if this is persistent in nature, if this will persist, then it will form the perisperm. It will form the perisperm of the fruit. So it will form the perisperm of the fruit or the seed, we can say. So the fruit and one more important thing is that the seed where the endosperm is present, those are considered as albuminous seeds. Okay, those seeds are considered as albuminous seeds. So in the albuminous seeds only we can find the perisperm, because that is the conversion of the endosperm. And if the endosperm is persisting, then only we can find the perisperm in such seeds. And if this is not persisting, then those seeds are considered as non-albuminous seeds. This is about the endosperm. What about the funicle? This is the funicle. So funicle will be converted into stalk. Funicle will be converted into the stalk of the seeds. In case of pea, let us take one example that in case of pea, we can find inside the pea there is this type of structures. Like this type of structures you can find inside the pea. So these are the stalks. These are nothing but the funicles which have been converted into stalk. Once all of these changes have occurred, the funicle will be converted into the stalk. This is considered as, this end is considered as the micropylar end. So what will happen to the micropyle? The micropyle will remain same. They will remain same. There will be no change. There will be one small pore in the seed through which they can get water, they can get air, so that it is helpful for them to germinate, so that they can germinate. That's why the micropyle is remaining same or as it is. And there is one concavity which is found in the seeds. Like this kind of structure we can find often. These are the concavity or a scar-like structure we can find. That is nothing but the hilum. So the hilum can give a concavity or the scar kind of a structure. Okay. The egg cell will be converted into the zygote and the antipodal cells. These are the antipodal cells which will be degenerating, as well as the synergids which are present there. These are the synergids. So synergids, antipodal cells, all of them will be disintegrating. Okay. Antipodal cell, synergids, all of them will degenerate. Actually, it is not the endosperm. Endosperm will give rise, endosperm, it is not endosperm. I was talking about the nucellus. Sorry, I was not talking about endosperm. I'm extremely sorry. It will be nucellus. The nucellus which was present outside this region, outside this embryo sac, there was the nucellus. If the nucellus is present, then that seed is considered as albuminous seed. and if the nucellus is absent, then they can be considered as ex-albuminous or non-albuminous seeds. And in most of the cases, the nucellus is absolutely used up by the growing embryo, and in few cases they are present. Like in case of castor, we can find the castor seeds, we can find that they are albuminous in nature. They have the persistent nucellus in them. So this is all about the post-fertilization changes which occur generally in the embryo sac and as well as for a flower. Okay, so in a lot of detail we are going to study the development of the embryo from the zygote. So keep on watching my next videos, and needless to say that if you found this video helpful, then don't forget to like, share and subscribe to my channel. Thank you.